Also okay to start now. And
Somebody's talking.
very good. I think New York is not logged in today.
Yes, he's on. Okay.
Okay, nie umiesz, okay.
Okay, let's start. You didn't miss anybody, right? All right. Very good. Okay, so quick recap. And Chris, you can join me. Yeah, you'll learn a whole lot.
Complicated.
Okay. Well, what I'm discussing is going to interest you a whole lot. Okay, so basically we're talking about lactose and the idea that, I don't know when they got dropped out. We can't hear you well enough. Okay, Chris, what are we going to do? I'm at 100%.
Oh, what's the question?
We can't hear you well enough, yeah.
Okay. Yeah, we're at 100. Our meter is going, so.
So Karen, we are at almost 100%. Our audio is really 100%. Did we have to change the type of audio transfer you
No, we were just doing that for
receiving their audio?
Receiving it.
Okay, all right.
So that should be able to hear us just fine. Can you
So Karen, can you jack up your
turn the volume up?
speakers? Yeah.
Yeah, we can hear you better now. Thank you.
Okay, very good. All right. Yeah, just double click there.
This one?
Yep. Well, you already know quite a lot. Okay. So, yeah, we were discussing lactose, for those of you, Wisconsin and Chelsea at Iowa. Lactose being the disaccharide is composed of the interglycosidic bond, which cannot be cleaved by alpha-glucosidase, which is the dominant energy monosaccharide producer in our GI tract. We have a specific enzyme that can hydrolyze lactose, that is lactase. And generally, as we age, the expression of lactase might actually go down. And therefore, if you consume milk or milk products, it could be cheese from pizza, by the way, that lactose accumulates in our colon, and the bacteria recognize this to be the source of energy, and in that fashion they try to get it from lactose. The way that you can hydrolyze this bond is by increasing the pH. If you increase the pH, and this is actually true in outside the body setting also, you can hydrolyze that bond by using strong acid. If you reduce the pH to somewhere in the region of 1 or so, then the hydrolysis becomes very efficient. And by the way, that particular technology of using acids to chop off glycans is the technology used in sequencing also. Okay. So you can actually do sequencing based on the same principle. But anyway, what happens because of the high acid is that lactose undergoes hydrolysis, but concurrent with that is the generation of highly acidic condition. So you generate acidity, at the same time you generate a whole lot of gases, and these gases kind of rise up, and this is one of the reasons why people have something of this sort. They have a lot of burping, and believe it or not, there are a number of hospital admissions that arise because of this simple problem, because a number of times people don't even know that they do not have enough lactase in the body. They suddenly generate a whole lot of gases. Concurrent with that is sometimes pain, and as soon as the thought comes that these gases or some pain is arising from close to chest, then the immediate thought is that I'm having a heart attack. That generates sweat, and people basically end up in ER. A number of times it is simply simple anti-acid that could fix the problem. So one of the things to realize, it's very important that if you have chest pain, obviously, to be very, very alert, but at the same time, remember that there are problems outside of the very dangerous ones that could also explain chest pain. Go ahead.
So even sucrose has a beta linkage, right? So is that also not going to be cleaved by alpha glucosidase, and would that also cause problems if we have too much sugar?
So sucrose is, let's see, sucrose is alpha-1,2 interglycosidic, so therefore it works really well. All right, very good. Excellent. And by the way, for Chris, you can see that many blacks suffer from something of this sort.
One of them.
Okay, very quickly, and this is primarily for those of you, and this is simply to stimulate your thought process. I know you already know the answer, most probably. Oops. You know the answer. Where do I go here? I go here. Okay. And point out the essential difference. What is the essential difference? The essential difference is
straight chain orientation of the or configuration of the C4 carbon.
that's correct. Configuration of, for example, the interglycosidic bond. So the interglycosidic bond out here is significantly different from the interglycosidic bond out here. That is arising because this particular sugar is significantly different from this particular sugar, and only in the orientation of that group. And likewise, this sugar, although it might not be evident because this sugar and both of these sugars are essentially in the same orientation, but at the C4 position, there is a dramatic difference, right? So for both of those residues, it is the C4 orientation that is different. And because of this, what happens is, because of this, the disaccharides are actually a swap. Okay, so this one is galactose, this one is glucose. This one is galactose, this one is glucose, right? So it is glucose galactose and galactose glucose. And you can imagine that both of these residues are going to be, both of these glycans are going to be significantly different, right? Because at least there are two positions at which they are different. And therefore the recognition is going to be different. You might be interested in knowing that, well, D-glucose is what is consumed by our body, right? It has to be D. D-glucose is sweet, so it is recognized by these receptors. D-glucose is metabolized. D-glucose is absorbed. But if you really look at L-glucose is much less sweeter, and it's typically not metabolized. So it basically can be thrown out of the body very easily, right? Very quick question. I don't know how many of you have heard me ask this question earlier. But if L-glucose is sweet, although it's less sweet than D-glucose, why is it? And the advantage is that L-glucose cannot really be consumed, right? So you can generate all the pastries of the world, all the ice creams of the world with L-glucose and have your taste buds really satisfied, and you won't generate any calorie because it's not absorbed. D-glucose, on the other hand, is absorbed. So we can put on a whole lot of weight. So why is that technology not used? Instead of that, we have aspartame, we have saccharin, we have so many other sweeteners that are used in products. But here is a very simple solution. Use L-glucose instead of D-glucose. Yeah?
Is L-glucose not naturally occurring?
L-glucose is not naturally occurring. Okay? D-glucose is. So what?
Is there an issue with accumulation?
Accumulation. Accumulation. It is involved in accumulation. Accumulation of what?
I'm asking if it's not an issue with accumulation, it being accumulated and becoming toxic.
In our body, it is left untouched. So it actually just passes through. It's not recognized. Yeah, you will have to use significant quantity. It is less sweet, so you'll have to use higher quantity. So you'll have to make higher quantity. It is not natural, so we'll have to synthesize it. And if you have to synthesize it, Can you actually compete with D-glucose in terms of the cost? Most probably not. Remember how many stereo centers are there in glucose? Quite a few, at least 5 plus one more, right? So there are quite a few stereo centers, and therefore synthesis is very challenging. Therefore, it turns out to be expensive. So this is not economically viable. Theoretically, the idea is good, but you can't really realize it in practice. And there are lots of ideas that are theoretically very good, but you may not be able to realize in practice. Okay, very quickly, I think we have gone overboard with the glycans, but is there a way to really, okay.
Move this?
Huh? Yeah,
Okay.
this is coming the way.
This was my audio, and let's bring this down.
Yeah, that should be fine.
Okay. Go back.
Simply to illustrate the structures that are generated, which are typically referred to as N-glycan and O-glycan. And I clearly don't have all the structures. The number of possibilities is huge, but there are two major, and we have a lecture especially on N-glycans and O-glycans coming up pretty soon. Okay, but this is simply sort of a curtain raiser where we can have N-glycan. So what is the meaning of N-glycan? The meaning of N-glycan is that there are certain residues in protein, especially asparagine. That particular residue in protein is connected with a sugar chain. And this sugar chain can be as long as 14 residues or longer. Or, it could be shorter than 14 residues too, but it could be significantly longer. And these sugars, or this is the glycan, generally when we talk to biologists, for most part, they are referring to glycans in this fashion, for most part. Okay. As I told you on lecture number 1, that even one residue is referred to as glycan these days by biochemists, which is glucosamine, N-acetylglucosamine. But generally, when people say glycans, it is the N and O glycans that they are typically referring to. Okay. Normally, you're going to see a stem such as this, right? What is this? This is glucosamine stem. Okay, mannose. This is mannose. And then you typically see bifurcation or branching. That's what it's typically called. There is a branch. And this branch has several residues. And likewise, there might be another branch coming off from here. So this branch, for example, is connected at position 6. This branch is connected at position 3. Right? Remember, this particular carbon is the anomeric carbon. Right? So there is directionality associated with N-glycan too. Okay? Got it? Okay. And generally speaking, as far as the N-glycan and O-glycans are concerned, especially the N-glycans, there is a certain block that is made all at once, and then it is transferred to the protein. And in the lecture on N-glycan, we're going to see that. That's called the N-block biosynthesis. But I want to point out few things which are very, very sort of overarching, and generally these are true for most N and O glycans, and I think I want you to remember those, right? So first of all, in this particular case, the N glycans they are fairly linear, but they do not have to remain linear all throughout. Generally, these are branch structures. Branching essentially means that there is one sugar residue on which there are two glycan chains that are connected. Okay? N-glycans means that the connection is to a protein, to an asparagine residue on a protein. Okay? And O-glycan means the connection can be to a serine residue on a protein. And there are other variants that are possible too. Okay? Right? So these are some very simplifying things. Now, generally speaking, you're going to see that the end residue on these chains, for example, here might have a residue of this sort. And this is not a 6-carbon unit. This is a 9-carbon unit, right? This is a 9-carbon unit. That 9-carbon unit is a sialic acid, uraminic acid of different types. Okay. So by the way, you don't have to have sialic acid, but you're going to find sialic acid a number of times. In fact, in some of the glycans, you're going to find more than one sialic acid. So you're going to find one sialic acid, then a second sialic acid, then a third sialic acid, and the chain could continue. So that is called polysialic acid termination. Okay? And there is a reason why that is the case. I'll ask you a very simple question. Now that you have understood about where these glycans are in terms of the structure, I'll ask you a very simple question. Why do you think that there might be, that nature might find sialic acid as the terminal, might want to use sialic acid as a terminal residue? Why do you think? By the way, there are several reasons. I want you to think of the simplest reason. You know what I'm asking? Go ahead.
Since there's a lot of hydroxyl groups, maybe it's easy to be metabolized.
If there is hydroxy group that can be metabolized, you mean to say there is a hydroxy group on the carboxylic acid position? Is that what you're saying? Where? Here?
Yeah.
So if there are hydroxy groups, they can be metabolized. Okay. Very good. Sugar metabolism does occur, absolutely. A little more complex answer.
Does it elicit like more specificity with protein interactions or something?
Okay, sialic acids are recognized, especially by microbes and so many other things, by immune system. So those things do play a role. Once again, very complicated answer. Bring it down to a very, very simple level. Very simple level. Because I do want you to think about, where are these, first of all, Q, where are these types of sugars present? They are present on proteins. These proteins are glycoproteins. A number of these glycoproteins are present on cell surfaces. Whereas sugars, in comparison to the amino acid chain in the glycoprotein, where is that location? If something was to approach, what would they come in contact with reasonably well? The sugars, because they are most extended. They are most extended outside. What is glycocalyx? Glycocalyx is a whole lot of sugars, different types of sugars, right? And glycocalyx is there. What does it do? It captures a whole lot of water. So likewise, if you have something of this sort and an ion, then the solubility in water is enhanced, and therefore it tends to retain water also more than simply normal sugars. Okay? So there is a slight advantage to having, simply from a physical perspective, not even physical-chemical, physical perspective. Okay? And then there is recognition that comes into play, obviously. Okay, what else do I want you to remember? So several points, right? O-glycan, serine, sometimes that is threonine. Asparagine is the primary residue. I do want to point out that this kind of branching is kind of unique, right? We typically don't see it in proteins. We don't see it in nucleic acid, right? This type of branching is kind of very interesting. Obviously, we don't really see in lipids, but the lipids can have in the glycerol, you could think of that as a branched lipid if it was glycerol. Let's not go there. Okay, any question? Okay, I do want to finish the section on structure. So let's quickly do polysaccharides, right? And here I want to illustrate some very simple points that I want you to take home primarily. Okay, so polysaccharides, there are a large number of polysaccharides in nature, right? Sugars are very, very common. In fact, all starches are basically all sugars. There are different types of starches, by the way. Okay? It depends on the composition. Sometimes the composition involves different types of interglycosidic linkages. most interglycosidic linkages in a starch is 1,4, alpha 1,4. Starch is something that we eat, right? We like a whole lot of potato chips. But it could be 1,6 also. And depending on the proportion, you could have different types. And starch can be hydrolyzed. Starch can be hydrolyzed by an enzyme. Right? We already said that alpha glucosidase is the one that does that. But then you can also have sugars connected in a completely different manner, which is called beta 1,4 linked glucose. So starch is alpha 1,4 linked glucose. Cellulose is beta 1,4 linked glucose. And as I was telling you in lecture number 1, that cellulose is not digestible. We can't really digest cellulose, and that's simply because the alpha has been changed to. We store polyglucose in the form of glycogen. Right? So if you eat too much, and the body recognizes that this is something that it might need in the future, especially during lean times, and therefore it turns on the machinery and simply stores it. So just because we are hungry, that doesn't mean that we should eat. Take it with a grain of salt. Okay? All right. This is cellulose, right? Very simple overview of what cellulose is and why it cannot hydrolyze. Okay, so I'm trying to give you a structural explanation of why cellulose is not digestible. So it's good not to eat paper or wood. Okay? But it's very good to eat starch or potato chips. This is cellulose. You recognize this? All of these residues are oriented such that this is glucose. And then that particular bond is in the same plane as that particular bond, right? And therefore the orientation of these two is beta. Right? And across the board you're going to see beta 1,4, beta 1,4, beta 1,4. So cellulose is a beta 1,4 polymer. On the other hand, starch, the more technical name of starch is amylose. Amylose is alpha. And here you can see that this particular, and this is also all sugar all across the board. And that particular bond and that particular bond are in opposite planes. And therefore, this is alpha. And in that fashion, this is alpha 1,4. So you can clearly see that even on simple hand draw structures, you can't really draw amylose and cellulose in a similar sort of fashion. Both go significantly different. The question is, why is cellulose a skeletal material, a structural material? Amylose is not a structural material. Amylose is something that provides energy, right? And the answer lies in this particular figure. And here you have to imagine a little bit. Okay? So I'm going to orient you once again. So for example, all the black balls are carbons, right? The oxygens are red and the hydrogens are white. Okay? And so this, for example, right, is connected to an oxygen. That carbon is connected to an oxygen. So therefore, this is the anomeric carbon. And this is carbon number 4, right? And this is carbon number 6. Okay? That bond is going down. That bond is going down. And therefore, these two are beta. And if you really look, what happens is, that is essentially like a plane. And this second ring is also like a plane. And likewise, this ring is also like a plane. And that ring is also like a plane. And these 4 different planes, because of the specific structure, can connect with adjacent rings through hydrogen bonding, because they are right in the same plane, right? Likewise, that particular is also connected, and it's essentially in the same plane. So therefore, you have one chain that goes like this, a second chain also goes like this, and they are exactly in the same 3-dimensional space, except that they're side by side. And now, you can have similar chains underneath, such that this oxygen, for example, can form positive interaction with that hydrogen. So now that hydrogen is now sandwiched between several oxygens. So you have one chain going in this fashion, There is a parallel chain, and therefore, if you have a number of those would form a sheet. And then you have a second sheet underneath that. And then you have a third sheet above that. So now you can clearly see that that essentially means that you have a structural material. Now, to penetrate this would take a whole lot of energy. So therefore, the enzyme cannot really penetrate something of this. So obviously, there is a recognition involved, but even if by simple means you have to penetrate this, it will take a whole lot of energy, right? So therefore, cellulose essentially is paper and wood and structural material. On the other hand, amylose or starch, which is the alpha, actually because of the orientation can only form, I was telling you just a little while ago, that when two units, they can rotate with each other. And so in amylose, 1,4, alpha 1,4, the polymer, they keep rotating, and because of they keep rotating, there is a helix that builds in. This is called an alpha helix, but actually this alpha helix is significantly different from what we see in proteins, right? The pitch and the rise are different. In any circumstances, this becomes a unit unto itself. It doesn't form sheets. It doesn't form these layers. And that is the reason why an enzyme can actually reach out to a certain segment and hydrolyze it. Okay? So therefore, this is digestible. By digestible, I mean you can hydrolyze it and the sugar can be absorbed. Okay? We have certain receptors that can pick up glucose. Plus, it's a small molecule. So therefore, there is passive diffusion that is also involved. Is this clear? Yeah? Okay. All right, I'm going to finish with a little bit structure of GAGs, glycosaminoglycans. All right? Glycosaminoglycans are fairly complex molecules. And the key point about glycosaminoglycans that I want you to remember is that glycosaminoglycans have a good number of sulfate groups on their residues. So there is a 6-sulfate, there is a 2-sulfate, there is an N-sulfate, there could be a 3-sulfate and various combinations. And we saw this in terms of structural diversity. When we were talking about structural diversity, we saw that. Now, because of these sulfate groups, what happens is, we already saw for amylose, for example, that it forms a helix, right? But also, when you have sulfate groups, that two sulfate groups, for example, that sulfate and that sulfate, when they come close to each other, that negative charge repel each other. And therefore, they tend to stay outward the most, as far apart as possible. So because of this, we have unique geometries that are created. So there is a helix, for example, this one and this one. These are two slightly different helices arising from something that I have not introduced to you as yet. Conformational changes, conformational variations possible in hyaluronic acid. But because of this, this entity is a sulfate, right? That entity is a sulfate. That entity is a sulfate. And so it tries to put it outside. Right? Two consequences. One, it dictates geometry as to what is stable. And number two, because the sulfates are on the outside, those are the ones that are recognized by the proteins with whatever that they interact with. Generally, primarily. I'm simplifying. But generally, primarily, sulfate groups are the ones. So if you don't have an appropriate sulfate and appropriate location, the recognition might be impaired. Not only that, the conformation will also change. And we all know conformation and structure determines function. Right? So this is, this is heparin. Right? Both of these structures are heparin. And this is an NMR-determined structure by Barbara Molloy in 1993. Now, heparin is just one type of glycosaminoglycan. There are quite a few types of glycosaminoglycans. So, for example, there is hyaluronic acid, or hyaluronate. There is dermatan sulfate. There is keratan sulfate. There is heparin. There is chondroitin sulfate. Those chondroitin sulfate can be of several different types: 6-sulfate, A, C, several different types of chondroitin sulfate. Dermatan sulfate is also called chondroitin sulfate B. Okay. If you look more closely, what you're going to see is the composition is different. The interglycosidic linkages are different. Okay. So hyaluronic acid has a fairly uniform structure. Okay. It's going to be glucuronic acid and acetylglucosamine, all connected with beta-1,3 and beta-1,4, two different types. Whereas if you look at heparin, out here, it's glucuronic acid. Doesn't have to be 2-O-sulfate. And it is glucosamine N-sulfate, 6-sulfate, for example. It doesn't have to be 6-sulfate. The connection here is alpha-1,4, okay? Alpha-1,4 across the board. And likewise, you can have different types of sugars that play a role. Now, all of these are polymers. They are fairly long polymers. Okay? The molecular weight could be as high as 50,000. Heparan sulfate could be as high as not shown here. Heparan sulfate could be as high as 150,000. So these are fairly long polymers. And with all the variations that are possible, I was telling you the other day, structurally this is fairly diverse. Okay, we have some idea about the structure based on not really X-ray structure, but an X-ray light structure. Okay, and this is published a long time ago. As of now, we don't really have too many structures of GAGs. Structures of GAGs are typically known from structures present as co-complexes. So GAG has to complex with a protein, and that has been crystallized, and we have some idea about those structures. And the deductions that I showed you earlier, for example, this kind of thing has been established for crystal structures also. So there is a reasonable alpha helicity that is known in the co-complexes of glycosomal glycan oligosaccharides with protein structures. Okay, so with that, I finished the structural part. Any questions at this point? Okay, very good.
Actually,
Yeah.
What about cellulose? So, you said that like on the glycosidic linkages you can have rotation. So, I guess if you were, looking at a piece of paper or something and you like zoomed in, at some point, if you just have like this linear chain of cellulose, like if one of the bonds just like rotate and it wraps around and it kind of just keeps going like that, because you said there's like hydrogen bonds between sheets too.
Between sheets, yeah.
Yeah.
So I don't really know. The energy required for a U-turn is very high. So you'll probably not have something of this sort. But it will basically terminate and then a new chain will start. Okay, let me check with Iowa and Wisconsin. What time is it? Should we take a break? 4:11.
Sure.
I want to take a 5-minute break. Okay. So we'll come back at 4:15. Okay?
Sure.
All right.
Thank you. It was great.
Yeah.
No questions.
Thanks.